Chapter 12, it's about the introduction to the nervous system. Nervous system itself is very complex, and it's the system where we lay foundation for our conscious experience, personality, and behavior. And neurobiology is a branch of science that combines behavioral and life sciences to study the nervous system. Both endocrine and nervous system helps to maintain internal coordination by sending signals, while the endocrine system uses chemical messengers known as hormones to send signals through a long distance. Nervous system uses chemical messengers that we call neurotransmitters to send signals between cells directly. Now, some of the functions of the nervous system is to sense the information that we receive from the environment and then transmitted that information into the brain and the spinal cord, which collectively are called the central nervous system. And then from there, the central nervous system will process this information, and then it will determine what appropriate response will be necessary to produce. And then the CNS will send a command or a signal into muscle cells or gland cells to make this command to happen. Central nervous system, it's a major subdivision of the nervous system, and it consists of the brain and the spinal cord, which both of them are protected by the skull and the spinal column. PNS, peripheral nervous system, is the nervous system or the cells, the nerve fibers that are located outside of the CNS, so outside of the brain and the spinal cord. And then within the PNS, we not only have nerve cells, but we have collection of neuronal bodies that we call the ganglions. The peripheral nervous system has two subdivisions that are called sensory and motor divisions, each which have a somatic and visceral subdivision. Now, the sensory division of the PNS is also known as afferent because it will bring signals from the receptors that we have within the different organs or different structures in the body, and it will send them into the CNS. Now, the subdivision of the sensory system will be called somatic sensory division and also visceral sensory division. So while the somatic sensory division will carry signals from the receptors located within the skin, muscles, bones, and joints, the visceral sensory division will carry signals that are coming from internal viscerae like the heart, lungs, stomach, and urinary bladder. So basically, somatic sensory division receives signals from all those major organs that are located outside of our body cavities. The other division of the nervous system is called motor or efferent, in which you have nerve cells that will bring the signals from the central nervous system into the effectors. And these effectors will be glands and muscles that will carry the body response. Now, as well as the previous subdivision, the somatic motor division will be carrying signal to skeletal muscles so that you can have muscular contraction, and then you can produce as well somatic reflexes like involuntary muscle contractions. And for the visceral motor division, which is part of the ANS, you will carry signals to the glands and the cardiac and smooth muscle, so you can have contraction of these muscles and then secretion of substances through these glands. And actually, this branch, the visceral motor division, it's under involuntary control, while the somatic motor division it's carried through the motor control. The conscious control. So another special subdivision of this visceral motor division is called sympathetic system and parasympathetic system. Now, sympathetic system, it's opposite in effect to the parasympathetic system, and this system, the sympathetic system, tends to arouse the body for action. We have a name for this division that we call it fight or flight division, because it will prepare you for situations that suddenly arise. And in this division, when you have activation, you will create an increase in the number of contractions of the heart. So you will have what we call tachycardia, and then you also increase the rate of respiration. So you will have tachypnea, and it will have a decreased effect on the digestive and the urinary system. So your digestive viscera will move less, as well as the distinct parts of the urinary system, or the different parts. Now, for the parasympathetic division, this is called also as rest and digest, as it tends to have a calming effect. In this case, you will have a slowing of the heart rate and breathing, and also it will increase the motility of the gastrointestinal tract and the urinary system. So in this diagram, you can see in blue the brain and the spinal cord, which forms the CNS, and exiting from the brain and the spinal cord, we have a different series of nerves, and also we have ganglia that form part of the PNS. So this is depicted in yellow. So here is a flow chart of what each of these subdivisions makes. So again, the central nervous system is formed by the brain and the spinal cord, while the PNS is formed by all the nerves that exit the brain or the spinal cord. And for the PNS, we have sensory division and motor division with their corresponding visceral part and somatic part. And the actual motor division has this visceral motor division that it is subdivided into sympathetic and parasympathetic systems. So the structural and functional unit of the nervous system are the neurons. And these neurons are excitable cells that respond to environmental changes. And these environmental changes, we call them stimuli. Once these neurons receive these stimuli, they will respond by producing an electrical signal that we call an action potential, and this signal will be rapidly conducted to other cells at distant locations. Now, these neurons can have this property also of secretion, because when they receive this electrical signal or this action potential, these action potentials will reach a part of the nerve that we call the end nerve fiber, and in this end nerve fiber we have vesicles filled with neurotransmitters that will be released, and then they will be sent into the next cell. Now, the neurons can be subdivided into functional classes. One functional class is called sensory or afferent neuron. In this case, these neurons will receive stimuli from the actual environment, and then they will send this information towards the CNS. Now, we have a second class of neurons that are called interneurons, that actually are located inside the central nervous system, and these will serve as a bridge between the sensory neurons, or also known as afferent, and the motor neurons that are also known as efferent. Now, the efferent neurons are also known as motor neurons, and then they will send signals to the muscle and the gland cells. So these will be their effectors. Okay, so here in this diagram, you can see the functional division of the neurons. So we have within our skin receptors that can detect several stimuli, like they can detect If a surface is rough or if it's smooth. It can sense changes in temperature. It can sense vibration. So we have different types of nerve cells in there and receptors within the skin. So when these neurons that we have within the skin receives the signal, this signal, it will be brought into the central nervous system. And this first neuron that you see here is called the sensory or afferent neuron. Now, within the central nervous system, especially within the spinal cord, we will have these interneurons that will form a bridge or connect. And as you can see here, these are short neurons, these interneurons in number 2. This will connect this sensory neuron that receives the stimuli and that it produces an action potential or an electrical signal, and it will bridge or connect to this motor neuron that you can see here on number 3. Now this motor neuron will have a command to be sent into this effector. In this case, it's going to be a muscle, or it can be a gland. So then sensory neurons are called afferent, while motor neurons are called efferent. And the neurons that connect them are called the interneurons. Now the neurons have different parts. One of the parts that actually bulk is called the soma. So neurosoma will be the control center of the neuron. Over there we have the nucleus, we have the nucleolus. Of course, within this we have the DNA, and we also have the cytoplasm that surrounds this nucleus. And then within this cytoplasm, as any other cell, we have mitochondria, lysosomes, Golgi apparatus, we have inclusions, and we have a rough endoplasmic reticulum and cytoskeleton. Now, one thing to keep in mind is that these neurons, they don't have centrioles, and they don't undergo mitosis. Now, emerging from the soma or the body, we have the dendrites, and these dendrites looks like branches of a tree, and their function is to receive signal from other neurons. Now, since there are many dendrites emerging from the soma, these dendrites receive more information that they can receive, and then they will provide a precise pathway for the reception and processing of the information. And then once this information is processed, this information is going to be interpreted by the neurons, and then they will send a response. And this response is going to reach an area of the neuron that we call the axon, which is actually an elongated part that is coming off from the soma. Now, the connection between the soma and the axon, or the beginning of the axon, is also known as axon hillock. Now, the axon is cylindrical and most of the times doesn't have branches, and at the end it has a part that we call the terminals. These terminals is where you have the storage of neurotransmitters. Now, the actual name of the cytoplasm within the axon is called the axoplasm, and the plasma membrane that protects and surrounds the axon is called axolemma. Now, many neurons have only one axon per neuron, while some neurons may have none, and many times these axons are covered by an extra layer that we call myelin, that it helps to transmit the electrical signals, or what we call the action potentials, much rapidly. So the axon terminals are little swellings that forms at the junction or synapse within the next cell. So here in this figure, you can see the different parts of the neuron. So this large part that you see here is the soma. This brown structure, round structure that you see here, is the nucleus. All these branches with these mini branches that you see here will be the dendrites. And then this long extension that you see here, it's an axon, and the beginning of the axon, or the connection between the soma and the axon, is known as axon hillock. So again, these dendrites receive information from the environment, they send it into the nucleus, the neurons will process this information within the nucleus, they will interpret this information, send a signal through the axon, and then this axon will end in fine branches that we call terminals. And this is the area of the neuron where we have the vesicles filled with neurotransmitters. Now the axon, as I mentioned before, is cylindrical, and it has this extra layer that it surrounds them. Not all the axons have them, but in this case this one does, that we call myelin, that it acts like an insulator. So this is this is a micrograph of the different parts of the neurons. So this is a neuron that you can see here. This part in blue is the soma, this will be the dendrites, and this part will be the axon that you see here. This is a picture similar to the one that we have in the home page for our course, and you can see here, this is the soma of the axon, this will be the nucleus, and again this will be the extensions or dendrites. Axon hillock at the start of the axons. Now the neurons can be classified according to their structure. So we have neurons that we call multipolar because they have one axon and multiple dendrites. This is the most common type of neurons that we have in the central nervous system. And as you can expect, since we have multiple dendrites, these neurons receive a lot of information, and they make a lot of connections with other cells through these dendrites. Now bipolar neurons will be the neurons that have only one axon and one dendrite, and such neurons are located within the olfactory tract that help us to detect the smells in the environment, and also within the eyes, in the retina. And as well, we have them in the ear, in a portion of the ear that we call the inner ear. Now, unipolar neurons will be neurons that have a single process that leads away from the soma. In this case, we have these neurons located within the skin and organs to the spinal cord, and these are sensory cells. Now, we can have some axons actually that they don't have we can have neurons that actually they don't have an axon, but they do have many dendrites. These are called anaxonic neurons, and we have them in the eye, in the retina, we have them in the brain, and also in the adrenal gland. So this is the structural classification of the neurons. So you can see, in this case, this multipolar neuron that has these dendrites in here, and then it has an axon in here, these two. We have unipolar neurons, so you have here this axon with these dendrites, and then we have these bipolar neurons in which you have two axons, 1 and 2, and then we have this anaxonic neuron in which you only have dendrites but no axon. Okay, so within the axons we have transport of materials, mainly will be proteins. Now, the function of this transporting of proteins is to repair the axolemma and also to produce within this plasma membrane of the axon, or to make, sorry, and introduce into the plasma membrane of these axons channels, and also to transport enzymes or neurotransmitters. Now, the axonal transport, it happens as a two-passage of proteins, organelles, and other material along the axon. So we have a transport that we call anterograde transport, in which you have movement of substances from the axon away from the neurosoma. And retrograde transport will be vice versa. So you have movement of the materials along the axon toward the neurosoma. So let me go back to show you about this transport then. So in this case, anterograde transport will be for the transport of materials from the soma into the terminals. And retrograde will be going from the terminals into the soma. Now, these materials that we transport within the axons are carried by a structure that we call microtubules. And within these microtubules, we have motor proteins that we know as kinesin and dynein that carry the materials on their backs, and they crawl along these microtubules. Now, kinesin will be proteins that are in charge of this anterograde transport, while dynein will be proteins that carries the substances in a retrograde manner. Now, the substances within the axon can be transmitted fast or slow. So fast axonal transport will be basically transporting substances between 20 to 400 millimeters per day, and for slow axonal transport, we'll be transporting materials between 0.5 to 10 millimeters per day. Now, in the case of slow axonal transport is always anterograde, while in fast axonal transport can be anterograde or retrograde. Now let's talk about the supporting cells of the nervous system, the ones that helps in the functioning of the neurons. So these cells that are supportive cells, we call them neuroglia. We have around 1 trillion neurons in the nervous system, and neuroglia outnumber the neurons by at least 10 to 1. So if you can calculate that number, that means that we have like 10 trillion neuroglial cells. And another name for these neuroglial cells are called glial cells. As I said before, they are supportive cells, and they help to protect the neurons and help them to function. So one of the functions then for supporting the neurons is to bind the neurons together and form a framework for the nervous tissue so that the neurons won't be floating. Now, in the fetus, they have an important function because they help in the migration of the neurons to their final destination. Now, if a mature neuron is not making contact with other neuron, it will be covered by the glial cells. And this will be preventing the neuron from touching each other and gives precision to conduct the neurons along a pathway. Now, the different glial cells are the following. So we have within the central nervous system oligodendrocytes, ependymal cells, and microglia. The function of oligodendrocytes is to form this extra layer around the axons that we call myelin. So they form myelin sheaths, and these myelin sheaths helps to make the transport of the action potential along the axon much faster. Now, the ependymal cells, they are located within the cavities of the brain, like the ventricles, and they help to secrete and circulate a fluid that helps to nourish the neurons that we call cerebrospinal fluid. Now, the actual ependymal cells are epithelial cells, and this type of epithelium is cuboidal, and they have cilia on the apical surface. Microglia will be wonder cells that are located within the CNS and that they look for debris and damage of the cells. So basically they will be considered like macrophages, and they have a very important function because any toxins of microorganisms or anything that it can damage the cells of the nervous system will be protected by these microglia. These microglia develop from actually white blood cells that we call monocytes, and then they become concentrated in areas of damage. Another neuroglia within the central nervous system is the astrocytes, and these will be the most abundant type of glial cells, and they cover the brain surface and most of the non-synaptic regions of the neurons in an area of the brain that we call the gray matter, which is where we have the actual body of the neurons. Now, these astrocytes have different functions, so they can provide a supportive framework so that the neurons won't be floating, and also they have some extensions of their cytoplasm that we call perivascular feet, that whenever they wrap around these capillaries in the brain, they form a closely tight barrier that we call the blood-brain barrier, or BBB. Another function of the astrocytes will be to monitor the activity of the neurons and to match this metabolic activity of the neurons to the blood flow that is going to go into these neurons so that they can have enough blood supply when necessary. Another function will be to convert a substance, glucose, into lactate and to supply it to the neurons. As well, astrocytes will secrete nerve growth factors, help to communicate electrically with neurons, and very importantly, will help to regulate the chemical composition of the fluid around these neurons, so it will help to absorb excess of neurotransmitters and ions. Now, astrocytosis, it will be the overpopulation of astrocytes when a neuron is damaged, and then the astrocytes will form a scar within the damaged neurons that we call sclerosis. So in this figure, we can see the different neuroglial cells of the CNS. So this one that you see here first on the left side on yellow will be the ependymal cells. Again, these are epithelial cells that are located within the ventricles, and they produce and absorb the CSF. And they have a cube-like shape, so these are cuboidal epithelial cells, the ependymal cells. And then here in green, you have an astrocyte. The astrocyte in here on this area is supporting this neuron and is wrapping on this area this capillary, forming the blood-brain barrier with these extensions of the cytoplasm that we call the perivascular feet. Now, in here on this side, we have an oligodendrocyte that it is myelinating this axon in here and this axon in here. Now, oligodendrocytes can myelinate more than one axon. And then in here, these cells that you see in red color, you have the microglia, what we consider the macrophages of the central nervous system. In the PNS, we also have neuroglia, so we have Schwann cells that, similarly as to the oligodendrocytes, they help to produce myelin and to insulate these axons. Now, the Schwann cells, they actually only myelinate one single axon, and they play a role in also assisting in the regeneration of damaged cells. Satellite cells is the other type of neuroglia, and these cells surround the neurosomas within the ganglia of the PNS. They provide also electrical insulation around the neurosoma and regulate the chemical environment of the neurons, similarly as to what the astrocytes do in the central nervous system. So this is a figure showing you a Schwann cell. So this will be the nucleus of the Schwann cell. This will be the axon in here, and these layers that you see here are the myelin layers that surrounds and protect these axons. This is another view of the Schwann cells. So the Schwann cells, when they are producing these myelin sheaths, they go around the axons several turns until they form a thick coat of myelin. And then the myelin later will be surrounded and protected by this connective tissue here that you can see that is called endoneuron. Oligodendrocytes at some point also provide several layers of myelin, similarly as the Schwann cells, as you can see here. So here it's a diagram of a cut of a nerve. So you can see that not all the nerves have this wrapped of or layers that surround them made out of myelin. So in this case we have unmyelinated nerve cells. Now, one of the functions then of the neurons will be to transmit information in the form of an electrical signal that we call the action potential. So we have different rates of conduction or speed of conduction of these signals across the nerve fibers, and this speed will depend on how thick is the nerve fiber. So when the nerve fiber is large, it will have more surface area and it will conduct the signals more rapidly. Now, another factor that affects how fast this signal travels through the nerves is the presence or the absence of myelin. Whenever a nervous fiber has myelin in it, the speed of conduction will be much rapidly than when they don't have this coat of myelin. So we have then these different fibers with different characteristics that send the signals, the action potentials along the nerves at different speeds. So we have a small diameter unmyelinated fibers that they can transmit the information between 0.5 to 2 meters per second. That will be too slow, huh? Now we have also small diameter fibers, but these will be myelinated fibers and they can conduct the signals between 3 to 15 meters per second. So as you can see there is a very huge difference or more speed when you have myelin whenever you don't than whenever you don't have a myelin in the small diameter fibers. Now large diameter fibers with myelin can have a conduction speed up to 120 meters per second. And yes, we don't have large unmyelinated fibers. We only have large myelinated fibers. Now, one example of when would you use a slow signal fiber versus a fast signal fiber is depending on the information that you are transmitting and from where it's coming. For instance, in the gastrointestinal tract, you don't need to transmit so much fast the information coming from the gastrointestinal tract. Thus, you use fibers that transmit the signal slowly. While in the skeletal muscles, where you need to improve balance or coordinated body movement, you have the transmission of the signal fast. Now, in general, the central nervous system neurons, they are not capable of regeneration. So once you damage these cells, they don't undergo mitosis. So that's why it's very important to take care of them. But in the peripheral nervous system, many neurons have the ability to regenerate. And if this occurs, it will happen because you don't have any damage to the body of the neurons or the neurosoma, and at least the plasma membrane within this neuron, which is called neurolemma, is remain intact. So you don't have damage to this plasma membrane. Now, some steps for the regeneration of the fibers are as following. So once you have this damage to this fiber, these fibers, the fiber distal to the injury will degenerate. And then macrophages will come and eat up this debris, and then you will have a swelling of this part where you have the damage, and then the ER will break up. So the endoplasmic reticulum will break up, and then the nucleus will move off the center. And since you have lost part of this neuron, you have an axon sprouting with multiple growth processes as the severe distal end continues to degenerate. And then Schwann cells will come in and they will start forming a regrowth pathway for the neurons to reestablish a synaptic contact, and then you will form what we call the regeneration tube. Now, once these neurons establish a contact with its original target cell, the neurosoma will shrink and it will return to its original shape or appearance. And then the nucleus also as well will return to the original space where it was, and then atrophied muscle fibers will regrow. In the case where a nerve cell that it was innervated, the muscle fibers were damaged. Now, regeneration is not fast, perfect, or always possible. You can have slow regrowth that may take up to 2 years, and some nerve fibers, guys, will connect with the wrong muscle fibers, and some will die. Others will regenerate of damaged nerve fibers in the CNS that it will not occur at all. So this is a diagram to show you then what happens after neuronal damage. So in here you have a normal fiber connecting, in this case, to a muscle, and then if this fiber it is damaged at this point of the axon closer to the soma, this part distal to the area of cut will degenerate, and then the macrophages will come and eat this part. Now, as this is happening, you will have regrowth of these parts of the axons. You have sprouts coming, and then the nucleus will move closer to this area, and then you will have secretion of growth factors that allows the reconnection of this neuron back into its target. And part of this will be because of the help of these Schwann cells that will start forming like a pathway so that the growth of this axon can happen in a specific manner without going into the wrong place. And then you have complete regeneration of the fiber, which leads to the nucleus moving back to its original position, and also for the shrinkage of the body as it used to be like the original cell was. Okay, now let's talk about the actual function of the nervous system, the electrical potentials and currents. So electrical potentials and currents are studied by electrophysiology. And this electrical potential, it's a difference in the concentration of ions from one point of the cell or outside of the cell from the inside of the cell. So you have a gradient. And then the cells, when they are resting or they are not performing any activity, the excitable cells like the neurons, they have a specific resting membrane potential that we call -70. So -70 is -70 millivolts. It's a negative charge inside the cells that are coming from these DNA, RNA, these proteins that have phosphate groups attached to them. And when you have activation of these cells, you will have a flow of current from the outside of the cell into the inside of the cell. And this flow of current is going to be given by the influx of sodium through sodium channels that we have within the plasma membrane. And then as this sodium is going into the inside of the cells, you will have a change in the resting membrane potential. Sodium, it's a positively charged ion, and as it's coming inside into the cells, it will make the cells less negative. So instead of having -70, you will reach a point of -60 millivolts in which your cells will reach a point that we call a threshold. Another name for resting membrane potential will be RMP. And this RMP happens because we have differences in electrolyte distribution between the extracellular fluid and the intracellular fluid. Now, ions tend to diffuse down their concentration gradient through the plasma membrane. And the plasma membrane is not open to the passage of ions just because they're ions. So it's selectively permeable. So only certain ions can pass. And we know that there is always electrical attraction of cations and anions to each other because cations are positively charged and anions are negatively charged. Now, potassium itself, it's a cation that it has the greatest influence in the resting membrane potential. Plasma membrane in general is more permeable to potassium than any other ion. And then we have these potassium channels that are always open within the plasma membrane, and then you have leakage of potassium outside of the plasma membrane. And then as this potassium is flowing outside of the cell through the plasma membrane, you will reach an equilibrium where there is no net movement of potassium. Now, if you let this to happen, you will find that potassium is 40 times as concentrated in the intracellular fluid as in the extracellular fluid. And then you have also within this cytoplasm of the cell these anions that will not escape the cells because they have a huge size and only small molecules can flow out of the cells. And then also many of these, they have a negative charge. So phosphates, sulfates, organic acids, proteins, ATP and RNA have a negative charge, so they cannot exit the cells. Now, the membrane in general is not variable to sodium, but the resting member potential is slightly influenced by sodium. Sodium is more concentrated in the extracellular fluid than in the intracellular fluid. And then when sodium leaks into the cell, it will diffuse down its concentration gradient. So since you have more sodium outside and less inside, it makes sense that it will go from more to less, or from outside to inside. And since you have negative charges inside the cell, in the cytoplasm, and sodium is positively charged, you will have attraction of sodium into the negative charges of the cell. Because of this influx of sodium, you will have leakage that makes the resting member potential slightly less negative than it would be if the RPM was solely determined by the outflow of potassium. Now, in order to maintain difference of sodium and potassium inside and outside of the cell, we have a pump that we call sodium potassium pump that will help to move sodium and potassium against their concentration gradient. So while the sodium potassium pump moves 3 potassium out of the cell, it will move 3 sodiums outside of the cell. It will move 2 potassium into the cell, 2 potassium ions. This sodium potassium pump requires ATP to work, and it does use 1 ATP per exchange. So you require energy to make this pump to be active, and 70% of this energy requirement is of ATP is required by the nervous system, because our nervous system uses a lot of glucose, and it uses a lot of this sodium potassium pump. Now, the nervous system not only needs glucose but also needs oxygen, and the exchange of 3 positive charges for only 2 positive charges contributes to -3 millivolts to the cell's resting member potential of -70. So in this case, we have these ions that flow across this plasma membrane through their corresponding channels, contributing to the RMP, or resting member potential. So in here we have the plasma membrane, we have the sodium channels, and in here we have the potassium channels. Sodium will be depicted as orange dots, while potassium will be depicted as blue dots. Now, since we have more sodium outside of the cell than inside, this is ECF, as you can see here, and this is ICF. Sodium will tend to flow along its concentration gradient because of that, so from more to less. And also sodium will be attracted by these large anions that cannot escape the cell, that will have negative charges. That's why they are anions. So we have here negatively charged molecules that will attract the positively charged sodium. And then we have these leaky channels for potassium that allow the escape of potassium along its concentration gradient. So from more concentration in the ECF into less concentration on the ECF. And then here you can see on this part the actual concentrations with their corresponding units in milliequivalents per liter. So we have for sodium 145 milliequivalents per liter in the ECF, while we have 12 milliequivalents per liter in the ICF. And for potassium, we have 150 milliequivalents per liter on the ICF, while we have 4 milliequivalents per liter in the ECF. So that's for the resting membrane potential. Now we have another type of potentials that we call local potentials. In this case, we have changes in the membrane potential of a neuron that occurs at a nearby part of the cell that it is stimulated. And then different neurons can be stimulated by chemicals, light, heat, or a mechanical distortion. And a chemical stimulant will bind to the receptor on the neuron, will help to open sodium gate channels, and then sodium will rush into the cell, and then you will have changes in the resting membrane potential in which the cell will become less negative, and it will reach a threshold that will lead to the more influx of sodium, so more sodium channels will open, and then you will gain more positive charges within the inside of the cell, so your cell become even more less negative, and this process is called depolarization that helps to reach the membrane towards 0 millivolts. Now, as sodium is entering into the cell, it will bring this flow of sodium that basically helps to trigger a zone in the cell that it will become more positive, and then you would call this a local potential. Now, for the local potentials, unlike the action potentials, they are graded. So that means that they vary in magnitude with the stimulus of the strength. So you can have strong local potentials or weak local potentials. And then a stronger stimuli will open more sodium channels. Now, we can have also what we call decremental local potentials, in which you have weaker then farther then they spread these local potentials from the point of stimulation. And then the voltage shift will be caused by the influx of sodium that will diminish with distance. And then you can have also what we call the reversible local potentials, in which if you stop the stimulation of these cells, the cells will quickly return to its normal resting potential. So these reversible ones will be weak local potentials because they don't spread at all. Now, the local potentials can be excitatory or inhibitory depending on the neurotransmitter that elicit this change in the area of these local potentials. So where they are elicited. So we have neurotransmitters that are excitatory, in which you can have depolarization of the cells, while we can have these inhibitory neurotransmitters that makes the cell less excitable, making the resting membrane potential more negative. So this is called hyperpolarization. Okay, so here we have a diagram showing you a part of the dendrite that receives a neurotransmitter and then allows the influx of sodium, creating a local potential. So in this case, if you magnify this and you see here the ECF and the ICF, and here you have the plasma membrane, you will see your sodium channels, and how upon binding of a ligand or a neurotransmitter into this area of the channel, you open up this channel that it was previously closed, as you can see here, and then you have influx of sodium into the cell, and then you can have this local potential. Now, action potentials are a dramatic change in the polarity of the cells that are produced by the opening of voltage gated ion channels. Now, these action potentials are generated only when there is a high enough density of voltage gated channels open. Now, the neurosoma has between 50 to 75 channels per square micrometer, and they cannot generate an action potential. But if you move a little bit away from the soma in the axon hillock, or what we call the trigger zone, you have many sodium channels or gates. We have between 350 to 500 gates per square micrometer. And then whenever you have influx of sodium within this area, this is where you can have the generation of these local potentials, and then as the sodium is flowing in, then you can have a stimuli that is strong enough that it will open all of these gates, and then you will generate an action potential. An action potential can be seen as a rapid up and down shift in the membrane voltage, and then it has different steps. So you have an arrival of a current at the axon hillock that will depolarize this membrane because we have a high concentration of sodium channels within this area. And then you will have sodium influx coming into the cell in this area, and then as the cells are becoming less negative, reaching the threshold point, which in this case in here it says minus 55 millivolts, but it can be minus 60. Then after this threshold is reached, you open all the voltage sodium channels, and then more sodium comes in, the cell will become less negative, and then it will become suddenly positive, and this is called depolarization. So once you reach 0 millivolts, sodium channels start to close, and then you reach a peak of the action potential of plus 35 millivolts, and no more sodium will rush into the cell. And then as this happens, as you reach plus 35 millivolts inside your cells, your your potassium channels will open, and then potassium will escape from the cell. And then your cell will start losing this positively charged ion potassium, and then you will return back the positive polarity of the cells of plus 35. Gradually you restore it into 0 millivolts, and then you go back into the actual resting membrane potential, which can be minus 70 millivolts, and this process is called repolarization. Now, as the repolarization is happening, you might reach the actual resting membrane potential, but you still can have potassium channels open that are open for a time, and then the cell will become a little bit more negative than the resting membrane potential, and this is called hyperpolarization. And then the astrocytes that are located within the area will start cleaning up the area from sodium leaks and also extracellular potassium, and then you will restore the polarity of the cell of minus 70. So here are then the steps of the action potential. So here you start In the y-axis you have polarity in millivolts, minus 70, minus 55, 0, and plus 35. And then here on the or on the x-axis we have time. So you start with the resting member potential of minus 70 millivolts, and then within the axon hillock, when you have an stimuli arriving, you start opening sodium voltage gated channels within the plasma membrane of the axon hillock. That will lead to the influx of sodium sequentially until you reach a threshold of minus 55 millivolts. As this happens, more sodium channels are open, you have more influx of sodium, and then your cell become less negative. This is called depolarization, and then you reach plus 35 millivolts, in which your cell is definitely positive. As you reach this plus 30 in here, or plus 35, you close these sodium channels, potassium channels will open, potassium will escape from the cell, and then you have gradually return of the polarity into every time less positive, and then you reach minus 55, the threshold, and then you go more to negative as more potassium is exiting the cell. And then for a time in here, after you reach the minus 70 millivolts, the resting member potential, still you have some potassium channels open, and then you have hyperpolarization of the cell, and then return back to normal, minus 70 millivolts. So again, everything starts with the opening of these sodium channels. Sodium rushes into the cell will become positive in here on this upper right panel, and then as more sodium is coming, your cell become more positive or less negative, however you want to see it. Then you close these sodium channels, open these potassium channels, and all the potassium that was inside of the cells escape, and your cell will hyperpolarize, as you can see here, and returning to resting member potential in your right lower panel. Now, these action potentials will depolarize only a segment of the plasma membrane. And then as this segment is depolarized, you will transmit this action potential into the following segment, the distal segment. And you will keep doing this over and over again until all the axon become depolarized. Now, once you reach a threshold in the steps of the action potential, you will have something that we call the all or none law, in which once you reach your threshold, you will have definitely the formation of an action potential. And then this action potential is not going to be decremental, so it don't get weaker with distance, and it will not go into this returning point. So you will have irreversibility, in which you go into completion and your action potential, it cannot be stopped. Now, we have also within this action potential some periods. We have a period that we call the refractory period that lasts few milliseconds after you have produced this action potential, And this refractory period means that you cannot elicit another action potential for as much as this stimuli that you are eliciting it will be. So refractory period will be the period of resistance to stimulation. And we have two phases. We have absolute refractory period, in which no stimulus of any strength will trigger the action potential, and this will last as long as the sodium gates are open, and then they become inactive. And then we have something that we call relative refractory period, in which only when the stimulus is super strong you can elicit a new action potential. And this will be actually when the cells are becoming repolarized. The returning of the positive charge into minus 70 millivolts, guys, is called repolarization. So in this case, your potassium gates are still open, and any effect of incoming sodium is opposed by the outgoing potassium. And then generally will last until you end the hyperpolarization. Now, something to keep in mind is that only a small portion of the neuron membrane is refractory at one time. The other parts will still be able to generate action potentials. So in here, this is what happens then during these absolute and relative refractory periods. So in here, you cannot have this action potential for as strong as the stimuli, and once you reach this part of the hyperpolarization, you can have a relative refractory period in which only with a strong stimuli you can form a new action potential. Now, when it comes to the conduction of the action potential, myelinated fibers, or fibers that do not have myelin, have voltage-gated channels all along their entire strength. So they will be transmitting this action potential throughout the plasma membrane. And they will be slow conducting fibers. While the action potential that it is transmitted through these fibers that are myelinated, they will conduct the signal much faster. Now, in the case of these fibers that do not have myelin, the action potential will be conducted all the way to the plasma membrane, and this is called continuous conduction. So, this is a part of an axon. Remember, the action potential starts here at the axon hillock, and then it will move from the soma, in this case, into the terminals. And as it's moving along into this unmyelinated fiber, you will have a continuous conduction or propagation of this action potential throughout the membrane. This is called continuous. And these colors that you see here represent different states of the action potential. So anything in green, that means that the action potential can be elicited. So these are excitable membrane portions in here. Anything in yellow is refractory membrane. So you may or may not produce an action potential depending if it's absolute or non-absolute refractory period. And then in here in red, we have the generation of an action potential, and then it will be conducted or propagated from proximal areas to distal. And if you see here, outside of the axon, which will be here, we have positive charges outside, and we can have negative charges inside. But in this segment, where you have actually the action potential in progress, you lose negative charges outside. So you have these, positive charges outside, you lose them. So that's why you have negative in here, and then you have positive in here. And as the action potential is moving, like as you can see here, the region behind this one that it was having the action potential will become repolarized. So in here you have return to -70, while this one will go into +35 and keep on going in that direction. So that's the propagation of the action potential, and it happens continuously within an unmyelinated fiber. Now in myelinated fibers, you have gaps in between the myelin sheath, and these gaps are known as nodes of Rambier. And in these nodes of Rambier is where we have these voltage-gated sodium channels that allow the passage of sodium, so you can have depolarization only within that area. And because only can happen then within these nodes of Rambier, and not in the action potential can happen in the nodes of Rambier, but not in the areas where you have the myelin, the actual action potential will conduct differently. So it will skip the areas where you have myelin, and it will go and propagate only in the areas where you have the nodes of Rambier. This transmission of the action potential within myelinated fibers is called saltatory or jumping. So here we have a myelinated fiber. Okay? If you magnify this region of the axon, you can see the insulation that this fiber will have, this axon. So in these areas where you have myelin, you don't have exposure of these sodium voltage gated channels. But in here, in these gaps in between these myelin sheaths, you have sodium voltage channels, voltage gated sodium channels, guys, and sodium can rush into these areas. These are these areas of unmyelination within these axons that are myelinated. These are called the nodes of Rambier. And this will help for the action potential to be generated in here and then migrate really fast internally and then propagate again in this area in what we call the saltatory manner or jumping conduction. So again, in here we have these axons, and we have different states of the action potential with the colors. So in here, in yellow, we have refractory, in which you cannot elicit or may, with the strong stimuli, the action potential. And red means that it's action potential in progress, while these excitable membranes are represented by green color. So in saltatory, again, you have this segment first depolarized, so you have the action potential in here, and then the depolarization follows like this, and then this area will be depolarized, sodium rush in, and it will keep on going in this direction, and then you gain this refractory period in here, and then in here where you have this refractory period, your cell become excitable again. Now, the cells of the nervous system connect to other cells in what we call synapses. And in this case, we can have a synapse between two neurons in the top part. And the first neuron that will send the signal is called presynaptic neuron. And this cell on the right side that receives the signal is called the postsynaptic neuron. And the direction of the signal transmission will be from presynaptic to postsynaptic. Now, in some cases, you can have connection between several neurons, as in this case, you have two connecting into this, and this one connecting into this other one. And at some point, depending on what area of the neurons these other neurons are connecting into, you would call the synapse differently. So for in this case, when this neuron, the presynaptic neuron, connects the neuron to the soma with its axon, which will be this one, you will call axosomatic synapse. And in this case, on the top part of B letter, where you have this axon of this presynaptic neuron touching a dendrite on this postsynaptic neuron, you would call it axodendritic synapse. And then in this case, where this axon of this presynaptic neuron is touching the axon of this neuron, you would call it axonic synapse. So this is an example then on the top of an axosomatic synapse. And this is basically a simple connection between two neurons, but most of our neurons, guys, connect to many cells. That's why we handle a lot of information, and that will be the case of panel B. Okay, so for the actual magnification of this area here, where the synapse happens. So this is what we will have. So we will have this presynaptic neuron. Okay, so you are magnifying this area. So this will be the presynaptic neuron with this axon. And the ending of the axon is called axon terminal. If you remove part of the plasma membrane in yellow to see the inside, you can see that inside the axon terminal, we have mitochondria, we have microtubules that acts like if it was a cytoskeleton, or well, actually it's a cytoskeleton, but acts like if you were the bones in our body to maintain a frame, so you don't lose the shape. So in this case, you have a cytoskeleton made of microtubules to hold all these organs and to have this shape of the axon terminal to be like what it is, like a button. And then we have vesicles filled with neurotransmitter. This neurotransmitter, once you receive an action potential, and then you have calcium influx into this area, these vesicles will fuse into the plasma membrane of the presynaptic cell, and then it will release through exocytosis. And then these chemicals will reach this gap that we call the synaptic cleft, and then the postsynaptic cell will have in its plasma membrane some channels that are chemically gated channels in which these chemicals, these neurotransmitters, will bind, and then these channels will allow the influx of certain ions. It can be sodium for excitatory neurons, or it can be chloride for inhibitory neurons or signals. Okay. So this is a scanning electron micrograph of many synapses within this cell. So all of these that you see here will be the terminals, and they are connecting to the soma of this postsynaptic neuron. You already have this slide. So this is a microscopic view. This is with light microscopy. So you have here this muscle fiber, and then in here you have this terminal of this motor neuron. And together, this connection is called the motor end plate. Okay, now let's talk about the neurotransmitters. So the neurotransmitters are chemicals, and they are classified differently according to their structure. The most common neurotransmitter within the somatic nervous system is acetylcholine, this one that you can see here, the one that makes us move our skeletal muscles. We have also amino acids like GABA, which is an inhibitory neurotransmitter. When we say inhibitory, basically it will make your neurons to be less active, or will be inhibited. We have glycine in here, aspartic acid, and glutamic acid. And in here we have excitatory neurotransmitters like epinephrine and norepinephrine, which are called catecholamines together with dopamine. Serotonin and histamine are also neurotransmitters. And then we have other neurotransmitters like neuropeptides, like encephalin and substance P, cholecystokinin, and beta endorphin, which is very low. When it comes to synaptic transmission, so your electrical signal that reaches these terminals now will become a chemical signal that move across the synapse. So in the case of synaptic transmission, you will have synapses that are different. Some neurotransmitters, as I told you before, will be excitatory, while others will be inhibitory. And these neurotransmitters will be having a different effect depending on the type of receptor that you have in the postsynaptic cell. Some receptors will bind to ligand-gated ion channels, and others will bind to proteins within the plasma membrane, and then they send a signal inside the cell through what we call a second messenger. Now we're going to see what are the different types of synapses. Excitatory will be in general cholinergic synapses, inhibitory will be what we call GABAergic synapses, and excitatory adrenergic synapses. So this is an excitatory synapse where you have release of acetylcholine. So when the action potential again travels or is conducted through the axon and reaches the terminals, you will have depolarization of this plasma membrane within the terminal. And through this depolarization, you open these channels in here that are voltage-gated calcium channels. So these channels will be voltage dependent. So only they will open if there is depolarization of this plasma membrane. So when the action potential arrives, you have depolarization of this plasma membrane. This calcium channel will open and then calcium rush in. Calcium is necessary for the binding of these synaptic vesicles into the plasma membrane within this area, and then is necessary for the release of this neurotransmitter. So it's necessary for the fusion and emptying of these synaptic vesicles. When this happens, you will have acetylcholine released into the synaptic cleft, and then acetylcholine will bind to these sodium and potassium channels. As sodium is rushing in, you will have depolarization of this postsynaptic neuron, and at the same time you have escape of this potassium. Now, our cells, as I told you before, when they release neurotransmitters, they release neurotransmitters in a huge amount, and these neurotransmitters cannot be always being stimulating this postsynaptic neuron, so they have to be degraded or recycled. So we have an enzyme within the synaptic cleft for these excitatory cholinergic synapses that is called acetylcholinesterase, or ACHE. When acetylcholinesterase binds to acetylcholine, it will break acetylcholine into choline and acetate groups, or acetic acid. So these acetate groups will be degraded within the synaptic cleft, while choline will return into the presynaptic neuron, so you can make more acetylcholine and then you start recycling. Now for the adrenergic synapses. In this case, you have release of vesicles filled with norepinephrine or epinephrine, and this norepinephrine or epinephrine will bind into the postsynaptic membrane, and they will bind as these receptors for norepinephrine or epinephrine, and then they will activate this G protein, which will be binding into this chemical messenger, second messenger that we call, or this enzyme, sorry, that is called adenylate cyclase. So when this G protein binds to this adenylate cyclase, you have conversion of ATP into cyclic AMP, and this will be the second messenger that will send a signal into the nucleus so you can have changes in the behavior of the cell, which it can be increase in the rate of transcription of the DNA, so you can have metabolic changes of the cell, or you can activate enzymes, or you can synthesize enzymes, and then you will have a response in which you open ligand-gated channels, and then you have a postsynaptic potential because you have entry of sodium. So at some point, this type of synapses works a little bit more differently than the previous one, and it takes more steps then for the depolarization of the cells to happen. Now, cessation of the signal will finish then when you don't have any more of this neurotransmitter available to bind into these receptors, and then your postsynaptic neuron will be returning into the resting membrane potential. Now, we talk about this presynaptic cell and these postsynaptic cells, and one thing that someone might think is that presynaptic cell, it only sends signals into the postsynaptic cell, so it's one-way communication. Well, in the nervous system, we can have both ways of communication. So the presynaptic cell will be the one that releases first a neurotransmitter, and then you have a signal coming into the postsynaptic cell. But the postsynaptic cell, once it does what the presynaptic cell tells it to do, it will send a signal back, and then the postsynaptic cell will become presynaptic, and the presynaptic cell, it will become postsynaptic. So there is two ways of communication. We have many neurotransmitters. We have around 100, but the most common ones will be acetylcholine, adrenaline or adrenaline, which are monoamines, dopamine, and also some neuropeptides that we already mentioned. Now, postsynaptic cells will undergo a potential that we can call excitatory postsynaptic potential, or EPSP, in which you will have depolarization and repolarization, or you can have something that we call inhibitory postsynaptic potential, or IPSP, in which you have hyperpolarization. And then, of course, in order to elicit this EPSP or IPSP, you have to have before an stimuli. Now, all these stimuli can add up for the excitatory postsynaptic potential, and then that will lead to an action potential in the postsynaptic cell because you have reached the threshold. Now, since neurons most of the times are receiving signals from many cells, they can do something that we call temporal or spatial summation. So in temporal summation, you will have intense stimulation from only one presynaptic neuron. In this case will be this one in here. And then as this postsynaptic cell is receiving these excitatory postsynaptic potentials, it will add them up, and then it will generate an action potential that will migrate towards the axon, or will be conducted in this axon. So this is temporal summation, basically adding up how many it receives on the same place. Now for spatial summation, you may have, in this case, all of these three neurons sending signals into these only postsynaptic neurons, so all of these are presynaptic. They are simultaneously sending action potentials into this postsynaptic neuron, and then this postsynaptic neuron is adding all of these action potentials coming from all these three presynaptic neurons, add them up into what we call a spatial summation, and then it will generate an action potential that it will conduct through this axon. Now we can have also within this communication between cells an event that we call presynaptic facilitation, in which you can have just nearby the terminal a connection from other neuron that it doesn't have no activity in facilitating the neuron, and then you don't have no neurotransmitter releasing here, and this presynaptic cell will have very minimal activity, and then this postsynaptic cell, because this one is not sending a signal, it won't be activated. But when this neuron receives an action potential, it will transmit these chemicals into this terminal, and then it will create an excitatory postsynaptic potential that, if you add them up with the signal within the presynaptic neuron that is traveling through the axon, it will change the voltage in here. It will make it positive, and then you will open these voltage gated calcium channels, and then this cell will become more likely to release the neurotransmitters, so it will be facilitated, and then this postsynaptic neuron will be excited. You can have the opposite. You can inhibit this presynaptic neuron, in which case it will be similarly as before. You will have a connection into this presynaptic neuron by this inhibitory neuron. This inhibitory neuron is not doing anything at all to this, and it allows only this presynaptic cell to only send a signal, or to keep sending the signal all the time to this neuron, and this neuron will be always doing something, the postsynaptic neuron. So you will always have influx of calcium, and this neuron that it is in here next by the terminal will receive a signal, and this signal will allow the release of an inhibitory neurotransmitter. So you will have in this presynaptic neuron an IPSP, or an inhibitory postsynaptic potential, which prevents this presynaptic neuron from releasing more neurotransmitter that will be otherwise always exciting this postsynaptic neuron. So you don't have more influx of calcium. So this will be inhibition. Now for the monoamines, which are epinephrine, norepinephrine, and dopamine. They are synthesized from amino acids by removing carboxyl groups while retaining the amino groups. Some monoamines are also histamine, ATP, and serotonin. These will be excitatory neurotransmitters. Purines like adenosine or ATP are also considered neurotransmitters. Gases as well are neurotransmitters like nitric oxide and carbon monoxide. And also we have these neuropeptides that we already mentioned, like cholecystokinin and substance P. Now, these neuropeptides are functioning as neurotransmitters or neuromodulators, and then they are also released from the digestive tract. So they are gut-brain peptides that causes food cravings. Cholinergic synapses are always excitatory, always uses acetylcholine, and they depolarize the cells, the postsynaptic cells, causing action potentials within these postsynaptic cells. GABAergic synapses are inhibitory. GABA it's gamma aminobutyric acid, and when this gamma aminobutyric acid binds into the postsynaptic neuron, it will open chloride channels. Chloride, it's an anion, so it's a negatively charged ion, so it will make your postsynaptic neuron more negative or hyperpolarized, making it less likely to become excited, so it's inhibitory. When it comes to these adrenergic synapses, these will work through second messengers, and they will have different response in the cells depending on what type of genes are activated by these adrenergic neurons. Now, adrenergic synapses are excited by norepinephrine. So in order to stop the signals, again, the postsynaptic neurons will stop receiving this neurotransmitter because it was broken down by enzymes like acetylcholinesterase in the case of acetylcholine, or because the neurotransmitter was reabsorbed by endocytosis like monoamine transmitters, or are broken down by an enzyme that is called monoamino oxidase. Or the neurotransmitter might diffuse into the extracellular fluid, or it can be reabsorbed by the astrocytes. Neuromodulators are also chemical messengers within the neurons, and they are secreted by the neurons and have long-term effects on a group of neurons. They help to alter the rate of transmission of neurotransmitters by the cells, also changes the release, reuptake, or breakdown of these neurotransmitters, and may adjust sensitivity also to the postsynaptic membrane to these neurotransmitters. Now, nitric oxide is considered a neuromodulator. In this case, this gas will enter into the postsynaptic cells, and we will activate a second messenger, helping for smooth muscle to relax. Encephalins and endorphins are also neuromodulators, and they inhibit pain signals within the central nervous system. Okay, so lastly, the neural signals are integrated. So we have to process and make sense of what these signals are, and we can produce an effect or store and recall information and use it to make decisions. Now, chemical synapses allow for this decision making. Brain cells are making a lot of contacts, like for instance, we have in the cerebral cortex the neurons that we call the pyramidal cells that looks like a pyramid. That's why they're called the pyramidal cells, the neurons. And they make about 40,000 contacts with other neurons. Because of this, you will have a trade-off. So you have chemical transmission that involves a synaptic delay. You cannot talk to all these these connections between these neurons. They cannot talk all at once. So this will slow the information as if they were no synapse. Now, the neural integration in the postsynaptic neurons is based on the postsynaptic potentials that are occurring in the cell receiving the chemical. Everything that reaches the threshold of minus 55 millivolts will be considered an excitatory postsynaptic potential, while in the opposite case, where you have inhibitory signals coming from these neurotransmitters like GABA, you will inhibit the postsynaptic potential in the postsynaptic cell because you have open chloride channels, and then you have exiting of potassium from the cell. So different neurotransmitters can cause different types of postsynaptic potentials in the cells they bind to. Glutamate and aspartate, they can produce excitatory postsynaptic potentials in the brain cells, while glycine and GABA produce inhibitory postsynaptic potentials. And then a neurotransmitter may excite some cells and inhibit others, depending on which type of receptors you have in the postsynaptic cell. For instance, acetylcholine and norepinephrine can be excitatory in some cells and inhibitory in other cells. Like in the skeletal muscle, acetylcholine is excitatory, but it is inhibitory in the cardiac muscle cells because they have different acetylcholine receptors. Anyway, so this concludes this lecture. Before we conclude this lecture, I'm going to give you the key points for this chapter. So the first key point is that the nervous system has different functions. So it can integrate sensory information, it can control peripheral effectors, it can coordinate voluntary and involuntary functions, and it can sense the external and internal environments. But it cannot direct long-term functions like growth, because this direction of the long-term functions like growth is given by the endocrine system. Now, the point of contact between a neuron and another cell, it's a synapse. That's another key point. Third key point is that the glial cells that can eat cell debris and pathogens within the central nervous system are known as microglia. The name of the axon's plasma membrane is axolemma, and when sodium channels open in the axolemma, you have depolarization. Now, when cholinergic receptors are stimulated, sodium will enter the postsynaptic cell. Now, autonomic centers within the nervous system can control blood pressure, heart rate, and digestion, but they cannot control skeletal muscle movements, because skeletal muscle movements are given by the activation of the somatic nervous system through these motor neurons. Now, the BBB, the blood-brain barrier, protects the central nervous system neurons from toxins, microorganisms, and drugs. And this barrier, the blood-brain barrier, or BBB, is made by the endothelial cells of the capillary walls of the central nervous system capillaries. A neurotransmitter will transmit information, and a neuron that transmits sensory information is known as afferent, while a neuron that transmits motor information is known as efferent. So this concludes the key points for this chapter. Hopefully you will be able to see it completely, and Hope you have a good rest of the day, or have a good night for some of you. Bye bye.